Well, good afternoon. I'm Dan Tarsy. The Movement Disorder Society has asked me to speak with Anne Young today about her career. I'm really looking forward to hearing some of these details. Anne Young to my left. And let me start by asking about, I know a little bit about some of what you've told me, but your early years as a student, for example, when did science sort of come up as something that you were very interested in, the scientific method, all of that? When did that first show up?
So I got interested, I think, because my parents were both sort of scientific groupies.
Mm-hmm.
They, one had done physics in college, majored in physics, and that was my mother, and my father had majored in chemistry. So we always had science and Scientific American, Nature, were always on our coffee tables, and as well as natural history. So those three or four journals actually were things that I'd start to flip through. And my parents never sat around the dinner table talking about, the weather or something like that.
Right.
They tended to talk about gravity or some phenomenon and how it would work and what would we think about it, how would we explore it more. And yet my dad was a businessman and my mother was a housewife. And I think my mother in particular had always been frustrated by not being able to be a scientist. She wanted to go to graduate school, was discouraged from going by her college. And she ended up actually working in the radiation laboratories at MIT as a computer, and worked afterwards, was asked to join the Manhattan Project and work there, but she got married and instead became
So did she have any role in encouraging your interest in science? Would you say there was a connection there?
Well, I would have to say that neither of them totally encouraged me. I had an older brother, and my older brother, they encouraged a great deal in terms of math and science and all of that. And I was competitive with him, so I sort of got myself interested in the biological aspects of things. And I just found it fun. We spent all our summers on a farm in Ohio, and on that farm, you didn't have much to do other than to go explore and things like that. So you'd go and check out all the animals. And I remember there were little ant colonies growing on our screens, and so I would watch them, herd the aphids around the ivy and all of that, and that just got me interested in that sort of approach. But I wasn't totally interested in science until I got to high school and I started to take some science courses, and that was very fun for me. So by the time I finished high school, I was pretty sure I wanted to be a scientist.
So the experimental method and ways of making discoveries as opposed to just simple observation, but actually intervening, doing things, creating situations, that started to get your attention?
Yes, and more, I think, in college than in high school.
Where did you go to college?
Vassar.
Okay.
And there weren't that many science majors at Vassar. So when I was a chemistry major, there were 9 chemistry majors in my class, and there were at least 9 faculty members in the chemistry department. So I had the run of an entire laboratory that I could, start to do experiments.
And did you become pre-med while you were at Vassar? You must have.
Yes, I became pre-med.
Yeah.
And I had not, however, taken freshman English because I can't read, as you know. And you had to read a book a week in freshman English, and there was just no way I could do that.
Okay, so you having said that, I think people want to know, what did you mean by that?
Right,
You can't
right.
read?
Yes, well,
Really?
I have a very hard time reading. I'm very slow. So I have to read aloud to myself in my head. And you just can't, verbally say the entire book in a week, or else I wouldn't be doing anything else. So I never finished any textbooks or anything like that. So I always learned by going to class and taking notes.
Was this at the time, given that period of time, was this recognized? I mean, were you considered to be dyslexic? Was that term even used back then?
No, I was not. No, my parents said that I was always, the teachers would always report to them that I was 2 or 3 grades behind my class in reading.
Yeah.
And I know that I could never finish any of the tests. And, but they were told it wasn't a problem.
Were there special accommodations made in test taking or examination taking? No.
None. No.
So what was your image of yourself at that time? I mean, did you see this as just a simple selective flaw in your makeup, or did you see it as a broader sort of...
I didn't even care.
Okay.
I didn't care. I was a little tomboy. I would pick fights with people. I was a tough little character. And I was tiny. I was skinny and I wasn't very big. But I could haul off and slug somebody really hard.
Wow.
And I...
you still do that?
The last time I did it was in medical school.
We'll leave that alone. So how did this, going forward, I mean, we're going to get to the rest of your career and so forth, but how did this influence your career? All the reading requirements, the things you needed to know about?
Right. Well, reading, I studied hard every night. And I also was one of these people that wakes up very early in the morning. I have a short circadian rhythm. And so I would do anything I had to do reading-wise, mostly in the morning. And I wouldn't finish it. I'd have to be sort of selective reading, bits and pieces of things. But the science, most people read science slowly. Sort of like I read everything slowly. People read science kind of more like that. So I could keep up with the science assignments. It's just that history I couldn't do, English I couldn't do. The ones where they'd give you a heavy reading load.
So that worked out well, since that's what you wanted to do anyway.
That's right,
Perfect.
exactly.
Okay,
right.
absolutely.
Right.
Then you went to medical school. You went to Hopkins. Tell me about that. Tell us about that. What was that like? What were some of the influences there?
Well, I went to Hopkins because I wanted to become a scientist. I mean, when I got out of high school, I wanted, or was in college even, I wanted to be a scientist. The whole notion of being a doctor to me was like weird. I didn't know what a doctor really did. And but my pediatrician was a friend of my parents, a personal friend of my parents, and he came to me and said, you really ought to do one of these MD, PhD things that are just coming in. And he said, there you can do the science, but your science then could be relevant to people, humanity, as opposed to bats and other things. So I thought about that, and I applied to the University of Wisconsin and to Hopkins, and I got into Hopkins. And then when I got there, I said, so can we talk about the MD, PhD program and how I'll set that up? And they said, well, you can do it any way you want to, because we kind of don't really have a program. We just wanted you to be here. You can get your PhD here and your MD. And so I went around and made a few deals with people. I went to it was at a time when at Hopkins they only required 1 year and 3/4 of science and 9 months of clinical rotations, and the rest was elective.
Wow.
So I went to the dean of the medical school and I said, I want to get a PhD, and I'd like to count all my elective time toward the PhD. Can I do that? And they said, sure, you can do that. And I said, can I get a letter to that effect? And
What made you ask for a letter? That's very clever of you to have done that. But
it turned
someone
out
give
to
you
be-
advice?
No, I just did it.
You knew it was the right thing to do?
Yes. And so I got a letter. Then I went to the head of the pharmacology department and said, I'm thinking of doing this in pharmacology. Would you count all of my med school courses in biochemistry and physiology and all of that towards my PhD?
Right.
And the head of the department said, yes. And I said, can I have that in writing? So I had those 2 letters, and I tucked them away. And that was a good thing, because I went through all the rotations I was supposed to at Hopkins. And then I started my PhD 2.5 years into the affair. And by the end of another 2.5 years, I had 13 publications and a thesis written. So they passed me on the thesis defense and everything, and then they came into me and said, it's going to look like we give out cheap PhDs at Hopkins. We can't let you get the PhD now. And I said, what do you mean? You told me that I could. And I said, I actually have these letters from you saying that I could do this. And they said, no, I don't think we can do it. We've changed our mind.
Couldn't do it at all? You'd have to wait a little longer before you got them back.
I'd have to spend another
Of the
year
year.
at Hopkins.
I see.
And I had already signed up for my internship in California. So I said, I can't do that. So I said, too bad for you. I'll just count it as a postdoctoral fellowship. And I'll tell every graduate student I know not to come to you because you guys don't hold your word. And I walked out of the room, slammed the door, and I thought I was finished for my PhD. And so I went down and talked to Saul Snyder, who was my mentor. And I said, I think I did something really bad. And he said, look, Ann, you just sit here. And he went down and talked to the room full of people, and they changed their mind. And,
The same
ended
day?
up okay. Yeah, the next day
Next
I
day?
learned. So, yeah, but it was one of those things that teaches
Interesting.
you.
So you mentioned Saul Snyder. He had a big influence in your early career.
Huge, yes.
Well, tell me, in what way?
Okay, so I got interested in the work he was doing, not only because he was a really superb teacher, but also a graduate student, a friend of mine, told me that the work he was doing in the laboratory was fantastic.
Was he a psychiatrist?
He was a psychiatry resident. At the same time, he was an assistant professor in pharmacology. And he had worked with Julius Axelrod, who subsequently got the Nobel Prize. And then he had come to Hopkins to sort of both do experimental pharmacology as well as psychiatry. And he turned out to be like a wonderful guy. He asked me to write a little proposal, which I did. And, we talked about the proposal, and he said, well, this is very interesting, but maybe you'd like to work on this. And gave me another project to work on. So I started working on that one, and it was going pretty well. And he taught me how you do the best pilot experiments. He said he had learned that from Julie Axelrod. Don't go making a giant experiment to prove your hypothesis until you've done a few pilot experiments to kind of gauge where it's going. And he was great at designing those in a way that would give you the most information with the smallest experiment. And if you had to learn something new or different, he'd always just pick up the phone and send you over to somebody else's lab to learn a new technique. He was always open to whatever you wanted to suggest. He was great.
So one of the things you got involved in was glutamate as a potential neurotransmitter. How did that come about, and what did you discover there?
Well, glutamate, I was interested in all the amino acid transmitters, glutamate, glycine, and GABA. And at the time, I was a graduate student. Many people thought that GABA was a neurotransmitter. They weren't totally certain about it, but nobody knew the main excitatory amino acid transmitter. It was the physiologists who were putting on small amounts of hundreds of compounds onto neurons and seeing what kinds of responses they were getting. And whenever they put on glutamate, they'd get an excitatory response. And when they put on GABA, they frequently got an inhibitory response, and same with glycine. And but nobody had been able to really prove that. And then what happened was, I had been going out with this other medical student, Jack Penny, who was rotating through a neurovirology laboratory run by Bob Herndon, Dick Johnson, and Les Weiner in another part of the hospital. And he came home one night, and we were just talking about what was going on, and he said, well, we have a virus that when it's you treat pregnant hamsters with this virus, it kills all the granule cells in the cerebellum without touching any of the other neurons in the cerebellum. So I thought that's a perfect way to find out the transmitter of those granule cells. And they were known to be excitatory. So in 4 experiments with the graduate student that was working in the virology laboratory, Mary Lou Ostergranit, and myself, we did these 4 experiments, and bingo, the only thing that showed up was glutamate was the only thing missing in those cerebella. And that was really, I think, the first biochemical evidence that glutamate was a neurotransmitter.
Were there any behavioral consequences in the animals that had their granule cells?
Oh, they were
away?
very ataxic,
Yeah.
yes. Yeah, definitely. Extremely ataxic, right from birth, you know.
And then I think you discovered it had something to do with elucidating glutamate's role in the corticospinal pathways and basal ganglia, not just the cerebellum.
Yes, ultimately, when I got out of my residency and got my own laboratory.
Okay, this was later on.
Yes, we started to work on, nobody in that interim period of 4 years had ever tried these similar methods to find the corticospinal transmitter. Most of the people in my field were in psychiatry, so they were very interested in dopamine, and opiates, and addictions, and all of that kind of thing.
So after Hopkins, you went out to the West Coast, San Francisco. You did an internship. And Jack went with you, or you went together?
Oh, yes. Well, Jack was a year ahead of me because we were in the same class in medical school.
Okay.
And then I took this extra year to get the PhD. And so he matched to California. And I went and applied and I'd gotten my internship, but then I applied for neurology residency. And the only place I wanted to go was UCSF. And it was in the days before the match.
Oh, yeah.
Well, let's put it this way. There was a match, but they didn't really,
They didn't call it a match?
They didn't quite call it a match. But at any rate, the people at UCSF were kind of worried because I had never done a neurology rotation.
Oh, really?
No, I never did one. It wasn't part of my 9 months required clerkship. So I had never done one, and they're going like, how can you apply for a neurology residency and you've never taken the subspecialty? I said, well, I've been working in neuroscience. I said, I know these guys, in the neurology department, like Dick Johnson and Bob Herndon, and they've taught me some neurology. I really like it. And so I think they were a little hesitant, but they took me anyway.
So you spent the internship, 3 years of residency
Right.
there at
Yep.
UCSF.
Yep.
And then comes the Michigan years. Is
Yep.
that, have I got that right?
Jack and I then had to look around for.
And when did you get married?
We got married in our second year of medical school.
Oh, okay.
Yep. In 1971.
Okay.
And,
So who did Michigan beckon to? Both of you?
Both
Jack?
of us.
Both of you.
Right.
You were a
had to.
team.
We had to sell ourselves as a package.
Uh-huh.
And so, we went around to places that had new chairs who might have several positions. And we got a couple of offers, but the best one was at Michigan, where they offered us a nice package. That was Sid Gilman. And he actually was a fantastic mentor. When we got to Michigan, even before we got to Michigan, I applied for a bunch of grants, and he went over them with me, he edited them with me, we sent them in. I got all of the grants I submitted.
Wow.
And actually one of the grants was an R01 that is interesting because the health science administrator on that grant was Nancy Wexler.
Oh.
She was working down at NIH. And Sid, I got a terrible priority score on my R01. And Sid says, you got to call up your program officer and ask them what you could do, what the key criticisms were, how could you get your grant better. So I called Nancy up. And Nancy, I didn't know her at all. I was scared to call. I felt like I was cheating, stuff like that. And she said, well, actually, your grant is going to be funded. And I said, what? I have a bad priority score. And she says, well, we decided to special your grant.
Wow. Why did you do that?
I said, why? And she said, because it was on interneuron activity and pharmacology in spastic paraplegic rats. And it turned out that they wanted this because of the VA and all these injured people from Vietnam and everything. They needed to have some research focused on spasticity. So they specialed my grants. So I got all these grants right from, I was so lucky. I mean, it was luck.
Right place at the right time.
Right place at the right time. Yep.
So, moving on, one of the things you're very well known for is your involvement in this whole, the circuits in the brain.
Right.
Cortex, the basal ganglion, back up again. Tell us about that. That's, I think a lot of people who read about that are a little unclear because of the authorship of some of the papers as to who the head honcho was in that whole thing. I don't know if it was one or a group, a collaborative.
Right. Well, okay, so this is interesting because Jack and I went to Michigan, and neither of us had done a movement disorder fellowship. Jack had taken a year of fellowship, but in neurovirology, and I hadn't done any fellowship. But I asked Sid Gilman if I could start seeing movement disorder patients. And the reason I asked for that was because, I was trained in neuropharmacology. In movement disorder patients, you're using all of these drugs that, I knew a lot about and felt I could manage perfectly well. And he said, yeah, fine, sure, hang up your sign. So we just hung up our sign, said we were experts in movement disorders. We knew
This
nothing.
was in the, what year was this now? This would have
This
been
was
by this?
1978.
Okay.
And
There weren't a lot of movement disorder fellowships, so
No.
to speak. Very few at that time.
right.
Right.
And we didn't take one, and we just hung up our sign, and we started to learn by seeing people. And one of the things that one of the ways that Jack and I worked together, right from the beginning, was that even as a medical student, he read for me, and then we talked together. So I never finished a textbook in medical school. He always read the chapters. He'd tell me, read this little section here, this section here, but we can talk about it. I'll tell you what, blah, 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 blah. And
He was a heck of a guy, huh?
oh yeah, he's a great guy. And he remembered all of it. He had a photographic memory, and I was just hopeless. So we would take long walks together. That's how we got our exercise, our bike rides, or whatever. But
Yeah.
we would be off together talking, and we would always talk about work. And say, how does this work? How does it work, that everybody says you're bradykinetic when you have Parkinson's disease. And you kind of see the patients tapping their fingers. But we'd say, actually when you watch them tap their fingers, a lot of times they get going really fast. That doesn't seem so bradykinetic. And, once they start walking, they actually go fast.
Too fast.
Too fast. Then they run into the wall. What's happening there? Then we started to say, well, if it worked like this, maybe this would happen. And we started making a little diagrams that we could say, look, if this went up, this should go down. And if this goes down, you're going to have a bradykinesia. If this goes up, you'll be hyperkinetic or whatever. And we looked. Jack was the anatomist. He knew all the anatomy, and I knew the pharmacology, all the transmitters, and how you would look at it. And from my work in Saul's lab, I had come up with a lot of thoughts about how you can look anatomically at receptors by sectioning the brain, soaking them in solution with radioactive glutamate or glycine or GABA or dopamine or whatever, and then rinse off the unbound stuff and put it next to a piece of film, and you get an image of where all the GABA receptors are in the brain, or where all the glutamate receptors are in the brain. And those images, you could actually do all the pharmacology with them, like you put in blockers and see what % blockade you got. You could look at association and dissociation curves, like how fast does it go on, how fast does it come off, all by using standards and different concentrations of radioactivity. And then Jack knew where to look for the anatomy to go in and say, oh, this is going up, this is going down, and so we looked at these issues and set up simple models first that I'd met Bob Moore at a meeting on spasticity, and I said, we've got this thinking about the basal ganglia. Would you be willing to read it? And we sent it off to him, this thing, and he said, I'd like it a lot. I'll suggest that it goes into the annual review of neuroscience. So we got that in 1983, but that didn't have the direct and indirect pathway in it. It had a set of pathways that we hadn't entirely explored yet, and in particular the output to the lateral globus pallidus versus the medial globus pallidus. And when we noticed over the next ensuing years, we had sort of put into the thing that dopamine was inhibitory. Well, then we'd get physiologists come to us and say, dopamine is both excitatory and inhibitory. And we'd go, come on, that doesn't make sense.
Depends where.
Well, it just doesn't make sense. They'd say in the same part of the basal ganglia, And I'd go, that doesn't make sense. And then we did the experiment where we lesioned the dopamine cells in the substantia nigra and looked at the output to the medial globus pallidus versus the lateral globus pallidus. Now, if dopamine had been inhibitory on both of those, then we should have gotten the same direction of regulation in both those palatal areas. But we got
The
totally opposite
Right.
effects in the two different areas.
Mhm.
Totally unexpected. So we're saying, there must be both an excitatory component and an inhibitory component to this. Then we started to look at substance P and enkephalin and these things, and it started to make much more sense.
Mhm.
And we said, it'll make more sense if we now go to Huntington's disease and scope out a couple of those pathways to see how that fits into the model, both in animals and in postmortem human brain. And that worked out nicely too. So we had these selective, we could then say that there was selectively different flow of information coming through these two different pathways. We didn't name them direct and indirect, actually, and neither did Malin. Another scientist whose name I'm blocking on now.
Strick or?
No, it was like McIntosh or something like that, but it wasn't McIntosh. He was a rat physiologist who brought up the notion of a direct and indirect pathway, and then we all started using it because that was a very nice way of phrasing it.
So where did Malin fit into this?
So Malin fits in.
This is Malin DeLong, of course.
Yeah, Malin DeLong fits in because he was doing work like this in the primate. And actually, Jack and I knew him pretty well, and we had a number of sit-down times. We did this, we went out and we specifically talked to Malin about his thinking. We came here to MIT, talked to Ann Graybiel. Malin, I can tell you, at least in 1986, could not explain his monkey data. I leaned over to him in the conference where some of this was being presented, and I said, I got the explanation for your monkey data. And then I gave my talk on the rat data. And he kind of goes, that makes sense. And because all we had is rat and postmortem human data. He had the monkey data, and his data were fabulous. I mean, and then his, he and his group were just fabulous experimentalists and made lesions in the subthalamic nucleus, and that made all the difference, I think. So,
Very interesting time.
yeah.
You mentioned Nancy Wexler earlier, and I wanted to sort of get into the Venezuela part of your career that you're very well known for.
Right.
And you spent how many, more than 20 years doing
22
this? Summers?
years.
Summers.
22 years, yeah. Oh, well, not summers.
Oh,
no,
not just-
usually in March.
Oh, in March, okay. And you'd go down there for a month or a couple of months? Or
No, I'd go down there usually for 3 weeks. And I was invited on the first trip that Nancy organized. There are a whole lot of funny things that happened. Nancy actually got her PhD at the University of Michigan, and
Her PhD was in psychology?
in psychology,
Right.
and she did it on being at risk for Huntington's. And
Which she herself was, is that right?
at risk for Huntington's, exactly. She was at risk for Huntington's. Her mother had it, and she got to know all the Huntington's families in Michigan. And there was a big clinic, and that's what I came to take over in 1978 because the guy at Michigan who had been doing it left and went to the Mayo Clinic. And I took it over, and the local chapter was suspicious of a new doc, and they asked Nancy to come up and check me out.
Oh, wow.
I mean, after all, I had had no training in taking care of Huntington's patients. So we hit it off. We liked each other immediately, and she invited me on the first sort of group trip down to Venezuela in 1981. And I went, my youngest daughter was just 18 months, and I was going off into what Nancy described as the jungles of Venezuela. And I was going to myself, ooh, that's a big, big ask, the jungles of Venezuela. But I was thinking to myself, this is it. Because in medical school, of course, what had I heard from Dick Johnson and Carlton Gajdusek? All about Scrapie and Kuru and all of those diseases and all their cool travels that they had done into remote areas. And I was thinking, wow, I could do that too.
You're going to become an explorer.
Yeah. So, and then I said, boy, but I don't know that I have it in me to do this. Could I actually go into the jungles? And then I said, if Nancy Wexler can do it, I ought to be able to do it. Little did I know that Nancy Wexler is like, she's fierce. Nothing frightens her. She would go into,
Really?
a black hole if she had to get the cure for Huntington's disease. So we went down there, and my part of the job was to go down there to this village on stilts out in a lagoon of the lake, 5 hours down from the main city.
This was Maracaibo?
Lake Maracaibo,
right.
Yep. And we lived down in this little town for 4 or 5 days each time we went down. And we'd sleep in hammocks on the porches of these little one-room stilt huts.
And what was unique about this group of people there?
Well, the reason for going there was that there was a man and a woman who were married, so to speak, and they had 14 children. And they both had Huntington's. So there was the opportunity, perhaps, to see somebody who was homozygous for Huntington's. And every other human disease that has been looked at, the homozygous of even a clearly dominant disease is worse. And in our case, in these 14 kids, there was some Huntington's, garden variety. And
so they weren't necessarily childhood onset or anything of that
No.
kind, just plain old Huntington's.
Just plain old Huntington's. And in fact, we found out after the marker and everything and the gene were ultimately discovered that, in fact, there were 4 homozygotes in that group, and you couldn't tell them apart from the others. And subsequently, that's been found to be true of other.
So this work sort of led initially, as I recall, the linkage data suggested a gene on a particular chromosome 4, was it?
Yep, chromosome 4.
And then a number of years later, the gene was nailed.
Yep,
So
10 years later.
10 years, wow.
10 years.
Did you think it was going to take that long?
No, nobody did. And it was a real eye-opener at how difficult it was. But what was also interesting is that Nancy put together a group, an international group of investigators that worked on yeast and worms and, every different species known to man, to say, what techniques do you use in your animal that would help us track these and crack the mutation? And it took this consortium, 10 years, and they looked at everything you can imagine.
So what did you feel like? What year was this now that the gene was identified? This is now
1993.
93. So what did that feel like?
Ecstatic, absolutely ecstatic. I mean, we thought we were going to have a cure pretty quick after that. And now it's 2015, and we still don't have one. But we're putting antisense in, and we're doing gene silencing, and that's pretty incredible. I don't think I would ever have believed that you could, first of all, get a substance that didn't cross the blood-brain barrier into your brain in concentrations that would actually decrease the amount of the Huntington mutant protein. And now there are techniques to do that. And so there's every reason to believe that, you know, if we get lucky, it'll work. So it works in animals.
Mm-hmm.
The other thing that Huntington's taught us in animals, because once we found the gene, we could make all these animal models. And the other thing that we learned was that you could put in the Huntington's gene and the animals would get sick like they had Huntington's, but you could put in a gene so that you could turn it on and off. And if you put in a gene that was turned on for maybe half its life, it got sick and its brain started to shrink and it lost certain neurotransmitters that are lost in Huntington's. And then you could turn the gene off and the disease would disappear and the animals would get better.
Wow.
Which nobody had ever really thought about happening in a neurodegenerative disease. That you could actually recover.
Reverse it.
Right.
Wow.
So that's what we need to set our sights on, too. in the
Okay,
future.
so there's hope for that.
Yeah.
I'm going to get back to something later, but as long as you're on that topic, there's a lot of excitement now about, we talked about this earlier today, neurodegenerative disorders of various types, Parkinson's, Alzheimer's, Huntington's, maybe ALS, in which misfolded proteins have been identified, different protein for each disorder.
Right.
Some discussion about prion mechanisms, maybe in Parkinson's disease. What do you think is the future of that going to be? You've seen a lot of these things mature, ripen, go in directions you expected, some going in directions you didn't expect.
Right, right.
How's this all going to pan out? We won't hold you to this. But
Yeah, don't hold me to it because I'm ready to be wrong as long as something gets right in the future. I think my feeling is that when the Huntington's gene mutation was identified and they made animal models, this occurrence of when they had, because they had antibodies to Huntington, they could stain the section, and then they saw the aggregates. And the amazing thing was that had actually been observed in 1979 by these investigators at Columbia University who had gotten approval to do caudate biopsies on living people with Huntington's disease. They got them out and they fixed them for EM, and they looked, and there in the nucleus were these fuzzy accumulations of something. But of course they didn't have antibodies to Huntington or anything, so they didn't know what this stuff was, and nobody ever pursued it. And it was, if only somebody had pursued it, we might have been a lot further along. But that's a little scientific lesson in and of itself. But then all the diseases, are showing these aggregates, Lewy bodies, amyloid, tau, tangles, even ALS has accumulations of proteins. But I think a lot of the data to date now would say that accumulation of proteins is already occurring after something bad has happened. That you're not handling the proteins appropriately before that even the aggregates actually develop. And that they're monomers or oligomers that can pass from cell to cell even potentially. And that's actually now been shown for tau, and alpha-synuclein does it, and amyloid does not. Huntington, there is some data that's arguably, needs to be repeated, that would say that there may be some transfer transsynaptically. But I think that those are very interesting diseases. It could well be that the neurodegenerative diseases are prions. I mean, as a clinician, you see Parkinson's and ALS sort of progressing from one limb to, down one side, over to the other side.
Yeah, I mean, it says Parkinson's remarkably begins unilaterally almost always. And why is that? And it's not related to the dominant side. It happens as if that's the first focus.
Right.
How it hits one side of the brain and then goes from there.
And it could be, you see, a prion phenomenon could do that.
Yeah.
And that gives you two more targets, drug targets. Target on the release side and the target on the uptake side on the other neuron. So
speaking of that, I was going to get to something else. You, in recent years, have had the opportunity to, as head of a major department and a research laboratory at Mass General, to work with a large pharmaceutical company by the name of Novartis. Did that for several years. What is that like? Because now the pharmaceuticals are jumping on this because this is potentially a blockbuster for sure.
Right.
If there's any commonality to these disorders, then you may have a real super blockbuster.
Right.
What was that like working with pharma, and are your goals are similar, but they're probably different kinds of influences. How does that work?
Totally different kinds of influences.
So say something about that.
Well, it's interesting because I put together this thing called MIND. And
what does MIND stand
really
for?
The Mass General Institute for Neurodegenerative Disease.
Okay.
And basically what it was we had researchers in Alzheimer's and Parkinson's and Huntington's and ALS, but their labs are sprinkled all over the huge MGH campus. And so I thought, they really ought to be sitting right next to each other, and they shouldn't be allowed to close their doors. So we built laboratories that had no doors on them, so you can't go in and out of the lab. You're just either in this huge set of benches, and Dr. So-and-so gets 3, and another doc gets 1, and other people get 6, depending on how many grants you have and everything. But all the fellows have a room where they sit all together, and you have to see what everybody else is doing. And then I gave seed money to people to say, look, when you're doing an experiment to try to find the ultimate cause of Alzheimer's disease or ultimate cause of Parkinson's, don't, along the way, you're going to have little branch points where you could take off and find a drug for that. But you're not going to be as motivated to do that because you won't have the resources, the expertise, or anything to do the screening and the libraries and medicinal chemistry. So at first I supplied some people to do that, and we got little libraries to do this on, 100,000 compounds or something, but not 1 million compounds the way the big drug companies have. But then we found some things and found some ways of measuring what we thought were key targets in these diseases, particularly Huntington's, and collaborated with Novartis. And they have very strict go, no-go rules.
Yeah.
And so if something doesn't look like a blockbuster in, time, 1 + 2, then boom, it's axed. And they don't, they don't think about it anymore. They just go on to the next thing. And that's a little different than we are, because sometimes we want to tweak the system, see whether we can tweak the drug.
Yeah, you want to keep at it till you get a solution.
Right. And then companies also, with Novartis, what happened was that the entire neuroscience division was discontinued, sort of in the middle of our collaboration. But we got to learn how the drug companies viewed the things that we did. And that was good because it allowed us, I think, to come forward with some compounds. Actually, in all the neurodegenerative diseases, we now have compounds that are in clinical trials based on stuff that was done at MIND. So, I think ultimately it worked, but you have to your collaborations with the companies are going to be very different,
Mhm.
and you have to try different ones. That's okay.
Mhm.
And the other thing is you have to get money to fund these things because the NIH doesn't particularly want to do some of those side projects.
Mhm.
They want to know the ultimate cause and fundamental science at the end. So they're hard to get funded. So you have to get that by.
I want to backtrack to something else, and that's about your. I've heard a little bit about your career, and it's how it was influenced by the fact that you're a woman,
Yeah.
and some feelings you've had about that over the years, and how you dealt with that. I mean, you
Right.
sounded like you're very forceful about getting these letters early on, but that
Right.
was that general principle of taking charge and making sure that you got what was coming to you, right?
Right, right.
So, but being a woman, what has that been like, and what do you think it's like now? Has it changed a lot? I hope it's changed for the better, but
Right, right. Well, that's very interesting. So I was just always just a tough kid.
Mhm.
And people said that I approached the world with my fists up.
this is
And
from when you were a tomboy on the farm?
yeah, and I kept that kind
Mhm.
of through. And I've always been pretty honest to the point where I've gotten into trouble by being
Yeah.
honest, by saying it the way I think it is. When I came to interview here, I thought it was a joke because for the job because at that time there were only 4 or 5 women who were like academic, had a research lab, and did clinical work, did in the teaching, and I ran an Alzheimer's center and blah, blah, blah. So I was asked to look at every open position in the country, when they had a search for a chair at this time period.
Mhm.
And it was just for affirmative action, and I would just write back, I'm not interested, I'm happy here.
Mhm.
But when they asked me from here, I said I'd never been to the Mass General.
Mhm.
So I said, sure, I'll come. And I came, and I was like blatantly honest with them. Almost like ridiculously honest with them. Well, I would do this, I would do that, I'd do it differently, department needs a lot of work.
Mhm.
And, I thought that will certainly mean that I won't come back. No, they asked me back. They kept asking me back. So then I had to get good advice about making a deal. And so I made up Jack made up the big spreadsheet for me.
Mhm.
All the costs, what it was going to cost me and everything. I got here, and I was the first woman chair of any department here. And
At MGH or
at
Harvard?
MGH.
MGH?
At MGH.
Okay.
And all the guys were so happy, quote unquote. And you'd be around a big table, and I felt totally comfortable there until the guy goes, "Do you feel comfortable here?" So, it's always funny because when you are the woman, you don't. I can't see myself out of my eyes, right? So I don't see myself there,
Right.
sitting there as a woman. I just see myself sitting there as a scientist and a doctor. And I don't take any bullshit from people. So that's the way I've dealt with it all my life. And there are jerks who are men, and there are jerks who are women. And what you have to do is you just ignore them. And the one thing I did do, which is a funny story, is that at Hopkins, all the lecturers, there are 9 women in a class of 110, and all the lecturers showed Playboy pinups during their lectures at Hopkins. And all of us would sit back, and the women would sit back and they'd look at these and we go, this is really ridiculous. And so one time this guy who had a little course where about a third of the class was in the course, taking a course on a chest x-ray. And he was sort of a little wormy guy. And he kept showing these pinup pictures and everything. And I noticed that he had his carousel, that he left it unguarded in the room before the lectures. So I went to the pathology department where I knew this guy and asked him if he could go down to the red light district in Baltimore and get me a full
the block,
frontal
right?
nudity of a guy.
Mm-hmm.
So I get this guy up on a cliff, all greased up, totally nude, and slide, and I slip it into the carousel. And the guy's giving his lecture. Up come the pinups and all of this, and then up comes this slide, and you can see panic on his face, and he clicks it to the next slide, and the guys in the class all go, "Back it up, Dr. So-and-so. The girls want to see this one, right?" So he's sort of forced to back it up, and it goes pale, completely pale. I thought he was going to faint. And the only good part was never showed another picture.
You cured him?
Yeah, I cured him. And I think I cured a lot of other people. It must have gone around somewhere. Somehow it got around that it wasn't. So my response to people is, you don't have to worry about, as a woman, if you're not treated properly, just get even.
Mm-hmm. The proper slide in the right place.
Right.
But you think it's gotten better?
Yes. So my advice is you don't have to get back, you just get even.
Sounds good. So, having said that, today, currently, a young woman comes to see you at some point in her training, critical decision-making time. What advice do you give potential women who are interested in neuroscience,
right,
and how to go about reaching their goal? Any special advice you'd give?
Well, I give the same advice to anybody that come to me. The first piece of advice is, and that is, do get it in writing. Mm-hmm. Because that actually, when I got those letters, I did not know that was going to make a difference. And I think that it's true throughout your career. If you come and you're recruited someplace, don't let somebody just say, this is what we'll have for you.
Right.
Always get it in writing. The other thing is, don't come in, women, if you're pushy, you're not viewed well by anybody. Mm-hmm. So, and certainly I tried never to be like pushy, but I would fight back if attacked. Mm-hmm. So, but I also think that women should, to avoid being called the B word, I think you need, and I tell them, to negotiate like Columbo. Peter Falk in thing, he would go, just trying to figure out how the knife got on the table. So you just go in to negotiate and you say, I'm just trying to understand how I can do 8 clinics a week and still write papers so that I can get promoted. I think that I'm wondering how my career would go. That kind of approach. And I think women, if they take that approach, will be viewed much more favorably than if they come in and say, I need this, I need that, gimme, gimme, gimme. I think that's true of men too, but men tend to be more forthright about what they need. Women, when they do that, aren't viewed as positively. And I think that's just a fact of life. The other thing I tell women is they have to ask, and most women do not ask. That's actually been a well documented now in the literature. There's quite a literature on it. Women don't ask. Don't ask for a raise. They don't ask for space. They don't ask for things for themselves. They'll fight like hell for the patient, but when it comes to them, they don't ask. and even my daughter, who's a neurologist now, she did, I taught her, and she doesn't ask. She feels uncomfortable.
So can I ask you, why do you think that is?
I think it's a problem women have. They feel like they're cheating in some way to ask or to.
Is this cultural
push.
or genetic or both, or do you have any?
I think there's aspects of both. Because there are some cultures where the men won't ask either.
Oh.
But that's unusual, and it's certainly not our culture. But I think there's also a gender difference. I think women feel okay, asking for other people, but they don't want to ask for themselves.
Interesting. One last thing. The Movement Disorder Society, this interview will eventually find its way on the website. You've had some contact with the MDS over the years.
Yep.
What do you think it plays an important role in this field?
Absolutely.
To accomplish something?
Absolutely.
What do you think?
Well, I think it has not only heightened the field, it's improved the science in the field. It has improved our care of patients. I think the journal has been fabulous. I think the meetings every year are really interesting. I think the videos that you get of all those patients are really very good learning material. And the journal did that very early on, and it was great.
I think it was the first journal to do video. Now there are several, but
yes, that's right. It was, and as I was telling you earlier, my article with Jack is in the first issue.
First one.
Yeah, and that was really nice to have. It was like a solid thing.
Thank you very much.
Yeah, thank you.
This was great.
Great.